What MCAT Biochem strategy helps with DNA repair? Imagine your DNA as a busy highway where mistakes and damage can happen at any moment. How do cells fix these issues so our genetic code stays intact? That's where understanding DNA repair strategies becomes essential for MCAT success. Knowing which mechanisms are involved can help you answer questions faster and more accurately. So, what strategies should you focus on? Let's break down the main ones and how they connect to the questions you'll see on the exam. First, think about the different types of errors or damage DNA can have. Mistakes during DNA copying, like mismatch bases, are corrected by mismatch repair. This process scans the newly made DNA strand and fixes errors that slipped past the proofreading activity of DNA polymerase. It's like a spell check for your genetic material. Then, there's direct reversal, which is used for specific damages like alkylated bases. Enzymes can directly undo these modifications without cutting the DNA backbone. Next, base excision repair is used when individual bases are damaged, such as from deamination or alkylation. Special enzymes called glycosylase recognize and remove the damaged base, creating a small gap. DNA polymerase then fills in the correct base, and ligase seals the strand. This process is like fixing a single typo in a sentence. Nucleotide excision repair is a bit more involved. It handles bulky lesions, like pyrimidine dimers caused by ultraviolet light. The system cuts out a segment of DNA containing the damage, then DNA polymerase and ligase work together to replace and seal the missing piece. Think of it as removing a bad paragraph and rewriting it. Finally, double-stranded breaks are the most severe. Cells repair these either through homologous recombination, which uses a sister chromatid as a template, or non-homologous end joining, which simply joins the broken ends together. These are like fixing a torn piece of fabric by sewing it back together or using a patch. For MCAT preparation, focus on understanding these mechanisms and the enzymes involved. Recognize that DNA polymerase's proofreading activity is a key player in correcting errors during replication. Knowing which repair pathway is used in a given scenario can help you quickly identify the correct answer. Also, remember that clinical questions often connect DNA repair to disease processes, like cancer, or treatments like chemotherapy. In terms of section strategies, practice questions that ask you to identify which repair mechanism is involved based on the description of the damage. Create mental checklists. Is the damage bulky or small? Is it a mismatch or a break? Which enzyme or process fits best? This approach helps you narrow down your options efficiently. By mastering these repair strategies and understanding their roles, you'll be better equipped to handle DNA repair questions on the MCAT. Plus, this knowledge forms a solid foundation for your future medical studies, where understanding how cells maintain genetic stability is essential.